friends, welcome back. In this video, we'll be solving GATE Excel MCQs and MSQs of Biochemistry paper 2024. In this video, we'll be solving 4 questions, each carry 2 marks. Let's solve. Question 40. Which one or more of the following statements correctly describes the fluorescence spectroscopy? Options are: the emission maxima is independent of the excitation wavelength. The emission maxima depends on the concentration of a quencher. The emission maxima varies with solvent polarity. The emission maxima varies with temperature. So the correct options are A, that is lambda max is independent of the excitation wavelength. Option C, lambda max varies with solvent polarity. Lambda max varies with temperature. Option A, option C, and option D are correct. Option B, lambda max is independent on the concentration of a quencher. It does not depend upon the concentration of a quencher. Let's move to the next question. Question 41. Which one or more of the following statements are correct in the process of pre-mRNA in eukaryotes? Options. Option A. 3' to 5' exonuclease activity is involved in the conversion of pre-mRNA to mRNA. Option B. 5' capping and addition of 3' poly-A tail precedes splicing. Splicing of pre-mRNA occurs via trans-esterification reaction. Option D. Alternative splicing can yield different mRNA products from the same pre-mRNA. So option B, C, D are correct. Option A, that is 3' to 5' exonuclease activity is involved in conversion of pre-mRNA to mRNA is incorrect because 3' to 5' exonuclease activity is for proofreading activity. It does not involve in conversion of pre-mRNA to mRNA. And option B, C, D. Option B, 5' capping and addition of 3' poly-A tail occurs before splicing in eukaryotes. It's absolutely true. Splicing of pre-mRNA occurs via trans-esterification reactions. There are 2 trans-esterification reactions of splicing of pre-mRNA. Alternating splicing can yield different mRNA products from the same pre-mRNA. It's absolutely true. So B, C, D are correct. B, C, D. Let's move to the next question. Question 42. Which one or more of the following statements correctly describes the changes upon the addition of puromycin during the eukaryotic translation? Options are: puromycin resembles the amino acid end of the charged tRNA. Puromycin occupies the A site of the translating ribosomes. Puromycin occupies the P site of the translating ribosomes. Puromycin occupies the E site of the translating ribosomes. In this question, what they're asking is, when there is the addition of puromycin during eukaryotic translation, what are the changes that occur? We have to select the correct statements from the below options. Puromycin is an antibiotic or a toxin which inhibits the protein synthesis, that is eukaryotic translation, and puromycin is a structural analog of 3' end of the amino acid tRNA. So when there is the addition of puromycin during eukaryotic translation, puromycin, due to its structural analog, it resembles amino acid end of the charged tRNA, and puromycin also occupies the A site of the translating ribosomes. So option A and B are correct. Option A, option B are correct. Puromycin does not occupy P site and it does not occupy E site. So option C and D are incorrect. Let's move to the next question. Question 43. Factor H, a complement regulatory protein in plasma, binds C3b and options are: competes with factor B to displace BB from convertase, initiates the catabolism of C3b into inactivate products, then binds to C3b BB convertase, acts as a cofactor for factor 1. The factor H, which is a complement regulatory protein in plasma, which binds to C3b and competes with factor B to displace BB from convertase, and factor H acts as a cofactor for factor 1. So option A and option D are correct. Here, this is the end of the video. Till now, we have been solving Biochemistry paper 2024. Till this video, all the questions from Biochemistry 2024, GATE Biochemistry 2024 is been solved. Thank you. See you in the next video.